Hello everyone, I'd like to welcome you to the first in this set of lectures devoted specifically to aspects of information flow with a bias towards information on DNA, RNA, proteins. This particular lecture is essentially meant to be devoted to the introductory aspects of the idea, if you like, the introduction to molecular biology. And these then happen to be the molecular aspects of what we'd call the biology and the new biology. These sorts of ideas form the basis very much of the features we also associate with the interpretation of data, interpretation of information flow. So we see this information essentially as laying that very solid foundation to the particular subsequent set of lectures you're now going to be getting. As you can see at in the first slide here, there's really an overview, if you like, of four main areas we'll be covering today. One of these happens to be the implications of DNA-based genomes. And so from that perspective then, of course, we'll be considering what the aspects are of what DNA is, how information flows, and what do we mean when we talk about a genome, if you like, the collection of DNA content that comprises, describes specifically the features of the information content of an organism. And from there we're going to go to the flow of genetic information. How is it that the information within DNA not only is copied but also carried through? And what you're going to see is that aspects of those involve the binding of key interactive components, particularly proteins, but also, of course, RNA. The RNA acts as an intermediate towards that information flow process. At the same time, we're going to recognize that there is no point in having a way to interpret information as it flows from DNA through RNA to the protein levels if, on the other hand, we don't recognize that there is a manifestation towards genotype and phenotype. In other words, what are the features, if you like, that describe those aspects of the appearance of both a cell, a tissue, and organism. And then finally, of course, we're going to be looking at what happens when some of these features go wrong. We're going to look at the molecular basis of two example inherited disorders. So what then happens to be the story here? Well, in terms of the first aspect of these, of course, we need to appreciate the implications of the structure of DNA. And DNA, as you will know from your previous reading, is essentially the oxyribonucleic acid. It's the genetic material. It's the main material we describe in most of the inherited forms being characterized at present. That's notwithstanding some other mechanisms of inheritance, but certainly that happens to be our bias. And so from that perspective then, we're going to concentrate particularly on the collection of inherited material that is DNA-based in terms of today's story. But please don't see that solely as one key solitary feature that stands as the single example. There happens to clearly be a whole range of additional ways in which information can flow. One of those, well characterized but certainly worthy of far more characterization, is RNA. The RNA-based genomes, particularly those found in retroviruses such as, for example, the AIDS virus. And so again, we recognize mechanisms of information flow. And there are other aspects of the course of information flow involving proteins. So let's then move on to considering what the features are in relation to features specifically of DNA. Now, insofar as DNA is concerned, on this next slide then, you can see I've presented for you what we can effectively describe as a minimalist viewpoint. The simple, idealized description of the content of DNA. And in this review of the story then, what we begin to recognize is that across the top, the components of DNA are not simply going to be the bases A, G, C, and T on their own, where A stands for adenine, G stands for guanine, C stands for cytosine, and T stands for thymine. But rather, as you can see, each of these components, adenine, guanine, thymine, and cytosine, as shown here, exist as connected up typically to additional entities. And those additional entities, of course, in this particular case, are sugars, deoxyribose. So you can see the deoxyribose connected in a very specific linkage. This 5-membered ring structure, deoxyribose, the sugar, again in each of these four cases. Now the first thing you can begin to notice over here is that the deoxyribose happens to, if you like, form a constant or common feature to all these four forms of connections. Yet at the same time, we also recognize that the 4 bases fall into 2 distinct categories. There are double ring structures, the purines, these are adenine and guanine shown across the top, and there are the single ring structures corresponding to cytosine and thymine, and those are shown across the bottom. Now, as many of you already know, I'll be subsequently describing to you how there is an important feature to this, which is double ring structures sit next to single ring structures in the overall structure of DNA. Well, let's get back to the major aspects of the slide here, because apart from the base, for example adenine in the top left-hand corner, and linked up to that common feature, the sugar, deoxyribose, which gives rise to the name deoxyribonucleic acid in DNA, apart from that, of course, the deoxyribose is additionally connected up to phosphate. And the phosphate, again, is a common feature. And that indeed describes for you some of the fundamental building blocks over here, such that when connected up in this way, we're looking at a nucleotide, or if you like, a deoxynucleotide, which in its essence then acts as one of the fundamental building blocks in the triphosphate form for the construction of DNA. Now, moving across the next slide, what you can see here is that, of course, the bases in themselves really are the way in which we convey information content. And those bases are really part of this larger structure that I've been alluding to. And that structure, of course, being the structure of DNA, clearly brings in this key feature of double ring structures next to single ring structures. And the more formal way to describe that from a chemical perspective, of course, is the purine sits next to a pyrimidine. So you can see here an example purine on the left-hand side, sitting next to a pyrimidine on the right-hand side. And this particular collection is guanine next to cytosine. And there are a number of ways you can know that, but I think the simplest way to see that is that there is a particular link in the way in which these are all particularly done, and that happens to be a very neat connection like pieces of a jigsaw puzzle. And there are 3 linkages shown here as a series of dotted lines between them. Those linkages are hydrogen bonds, a kind of a weak glue between these. And I think a wonderful way to look at the story is to imagine that you happen to have the purines, either A or G, or the pyrimidines, C or T, and you shuffle them around on a desktop. Cut out versions of these in the classical way, and there's a wonderful story behind this that involves Watson and Crick. As this is then shuffled around on the desktop, what becomes immediately apparent to you is that you can place the purine and the pyrimidine right alongside each other in this very neat way to allow this gluing process to take place. And the other neat thing over here happens to be the connection points, although not shown here, for the sugar, the deoxyribose, in both these cases comes off the bottom left or the bottom right of that figure. So what's beginning to build up over here is that you happen to have a very specific jigsaw puzzle connection between the G and the C in this example. We also recognize, of course, that there is A and T involved in the story here too. And the A and T also, if you shuffle them around, place them next to one another in the same kind of way, that not only do they form a very neat connection like these pieces of the jigsaw puzzle, but that in addition to that, they form hydrogen bonds. But in this case, only 2. So the glue between A and T is weaker than the glue between G and C. That's a weak glue, a hydrogen bond-based glue, but it also tells you now, of course, that it's much easier to separate the A's and T's compared to G's and C's. That's relevant to what we'll be discussing a little bit later on. The final part of the story here is that if you place the G and C base pair, as we can now call them, because it's a pair of bases, if you place the G and C base pair down, we now take the A-T base pair and place it down as well. We can see they have virtually the same external dimensions. So the distances on the outside of the A-T base pair and the G-C base pair are essentially the same. Now, when we start to recognize the relevance of this, it takes us through to the next slide. And now what you can see is that DNA essentially consists of a series of connected bases. So it's now no longer the case for us to consider an AT base pair or GC base pair. Rather now what we need to consider is, or focus upon, is